Media is what the recipe is. So what's the chemical content of what we're actually putting in with the agar? So we have our seaweed agar part, and then what is the other stuff that we're adding in that the particular bacteria likes or doesn't like? So the two main types that we have is defined or synthetic media and complex media. Defined has a very specific chemical composition. So it's a recipe that is highly repeatable. It's going to be the same if I make 200 plates, all of them should be identical. Complex media is media that contains extracts from animals, plants, yeast, things like that. So it would be like I often use sheep blood agar. There's bovine brain agar, we have casein milk agar, things like that. It's really hard to get like a milk agar to come out identical every time. It'll probably be very close, but it's not nearly as specific as a synthetic or defined media would be. Okay, so some examples here. This is a defined or synthetic media. And to make this, you would go get your scale out and go, I'm going to have 0.25 grams of all of these amino acids or subgroups of a protein. And I'm going to need 0.5 of these amino acids and 0.12 of these ones. And I'm going to add exactly this much of all these vitamins and these salts. And I'm going to buffer it, and it's going to be the same every single time. Whereas if we look over here, this is the complex media, and we're going to go, okay, 27.5 grams of brain extract and 2 grams of glucose. Well, brain extract or heart extract, those aren't going to be the same every time. We may be weighing it out, but they're not going to have exactly the same protein composition. Hence, this one is a more complex media. The other thing that defines which media we choose is what our purpose is or why we're using it. So we have general purpose media. This is media that allows the growth of a wide range of different microbes. Basically, we'll let almost anything and everything grow. The main one that I use for this is tryptic soy broth. Pretty much anything will grow on it. Like if I leave the lid off in the lab for an hour, come back the next and put it in the incubator, come back the next day, a whole slew of different looking colonies are going to be growing. Pretty much anything and everything. Enriched media is media that contains complex organic substances. So again, that would be like our last slide where maybe we're adding blood or special growth factors to it, all with the goal of getting some species to grow and not others. So maybe we have a blood agar down here. Maybe we're going to put our patient sample on it, and we're only going to collect the colonies that make little clearings here in the red agar to find the ones that can digest blood because our patient has some sort of blood disorder, okay? Or maybe we're going to be thinking about what growth factors are important. So if we want to see which microbes need which amino acids or vitamins, we could also have that goal. Bacteria that require very specific growth factors like different vitamins or specific complex nutrients like blood or milk are known as fastidious, basically high maintenance bacteria that we have to work really hard to get them to grow, to culture them, to figure out what they are. One example of this is Neisseria gonorrhea, which as the name suggests is the bacteria that causes gonorrhea. And to grow this, it has to be grown on a chocolate agar shown up here. And no, it doesn't have chocolate in it, even though it looks brown like chocolate, I know. It's a enriched media that has blood in it and a whole bunch of other nucleotides in it that are specially cultured to grow only Neisseria gonorrhea. so that if you put a patient sample on it, nothing grows, then hey, no Neisseria gonorrhea. But if we do get these white colonies, then we know that that's the pathogen that we're dealing with. Enriched media is useful in the clinical setting because it will encourage the growth of some pathogens, even if there aren't very many in the original sample. And so another tool kind of similar to all of this is selective and differential media. So selective medias are agars that we have made that will inhibit the growth of certain bacteria, but encourage the growth of others. So if you have a patient sample that is a mixed culture, we don't know what's in it, but we want to determine which one's the pathogen, then we could put it on a selective media and it will inhibit some bacteria and only allow others to grow. So an example here is maybe you're concerned your patient has an intestinal pathogen. You could use a McConkey agar, and it has bile salts, which again would be something you would see in the intestines. You would expect an intestinal pathogen to need bile salts, but maybe a skin pathogen to not need it, okay? And so we could put the culture on it and only the intestinal pathogen would grow and not anything else. Differential media is media that will let anything and everything grow, but it has a color change that tells us something. So in this example, same agar, it's both technically, okay? McConkey agar will differentiate between whether the bacteria can use lactose, ferment it, or not ferment it. So it will often use a pH indicator. And so if the bacteria can do whatever it is we're testing, it'll turn red. And if it can't, it'll be yellow. Or another example would be, I have some agars that look for fecal coliforms or gut bacteria that can be found in contaminated water supplies. Okay, so we'll look, and if there's fecal coliforms, they'll turn purple, and the ones that are just regular old soil bacteria will turn white. Okay, so it's a color change, but all the bacteria will still be growing. It just tells us something about its metabolism. Okay, so more examples of selective media, medias that only let some grow and not others. So they'll use things like tomato juice and acid here. It will only let lactobacillus in saliva grow. Okay, so other bacteria, if E. coli is there, it's not going to grow. Okay, so you can use a whole bunch of these different ones if you have a hunch that your pathogen is dealing with, I don't know, too much lactobacillus in their saliva, or if you think, oh, salmonella is the problem, well, maybe you'd use the Salmonella shigella agar. Differential media will let everything grow. It just has a visible change on the agar. So things like maybe you're looking for the triple sugar iron agar. Okay, well, if your bacteria can ferment the sugars, then they'll make a sulfur product and then the bacteria will turn black. Or maybe if you're looking for hemolysis, you'd use a blood agar. It has blood within the agar plate, and based on whether it can clear out the agar or eat the blood in it, then you can determine, okay, what sort of pathogen are we working with? Okay, so again, to reiterate, we have our mixed samples up here. If we put the mixed sample that has multiple types of bacteria in it, we want to know what's in there. If we put it on a general purpose media, everything's going to grow. They're all going to look vaguely the same. And then, but if we put that on a selective media, for maybe we're looking, can it ferment lactose? Then only the bacteria that can ferment lactose would grow. Everything else would be inhibited. Or if we take the same sample, again, general purpose media, they're all going to look vaguely the same, but maybe we put it on the blood agar, then we can go, okay, well, all of them are going to grow, but they're going to maybe be different colors or have visible differences in what they do to the agar. couple more kind of miscellaneous medias we can use. We can use reducing medias that contain substances that will absorb oxygen or slow the oxygen that's available. Okay, so this can be important if you're studying what sort of gases your bacteria needs. We can also use carbohydrate or sugar fermenting medias. These contain different sugars. It could be maltose or sucrose or glucose. And if your bacteria can use that sugar and they can ferment it, it leaves a byproduct that changes the pH, which we can't see pH change, but we can if there's a color change indicator shown over here. And you can go, okay, well, if they're fermenting it, then maybe the starting red will turn yellow, or if they're not fermenting it, oh, it's still going to be red. We also have transport media. This is media used to preserve specimens. So maybe you take a patient sample at the clinic and you take their blood sample or a stool sample, something like that. And your clinic doesn't have a lab on hand. You have to go across town. So you'd put it in the media to kind of freeze it in time to preserve it. And then you can run it across town, bring it to the lab, and it still has all the microorganisms intact. It hasn't deteriorated. We can also use assay media. This is media used to test how effective different drugs are on that bacteria. So trying to determine which antibiotic, which antimicrobial is proper to use on that particular pathogen. And we have enumeration media. So this is used to count the number of microorganisms in a sample. This is critical for water and food testing. So if we want to know how many fecal coliforms are on your kale from Whole Foods, well, you'd use an enumeration media. Or if you're trying to study how many bacteria are in Lake Evelyn, is it contaminated or not? Okay, things like that, you'd use the enumeration to count how many did we start with in our original sample. Okay, so once you've grown it all up, we're on to isolation. This would be separating individual colonies from the other cells. Again, a colony is that discrete mound, that circle of cells grown on the agar, and this indicates we have one individual species of bacteria. Okay, so let's follow the little chart here. We have our mixed culture. It has, I don't know, our red rod-shaped bacteria and our circular white bacteria. And in the tube, I can't tell you the difference. I know they're probably both there, but can't really tell. Okay, so we're going to separate them by putting them on the agar. We're going to spread the agar around over here. And as they grow up here, we're going to get that one white colony is going to turn into 10, 100, so on. And we're going to get our little mound of white colonies and our mound of red ones. And they're discrete enough that with my loop, I could take just the one red colony and not touch the white one, or just the white colony, not the red one. Again, from the broth, can't really tell by eye, still can't tell when I inoculate the plate, but once they grow up and we see the individual colonies, I can pick them individually and isolate them. There are a couple ways to inoculate a plate. And the first one is a streak plate. So we would take our loop here, and with our original sample, it's been all sterilized. We take our sample, we would spread it back and forth over here. I'd flame it again, if you pretend my mouse is the loop, and then I'd streak it over here, flame it again, streak it over here, flame it again, streak it again. The whole goal of doing a streak plate is to dilute your original sample. Okay, so that's why I'm not adding more each time I streak. I'm just spreading from where I started at. This basically hedges your bets. So if you had like 5 bacteria to start with. If you spread in this first quadrant, you're going to get something to look at. But maybe you started out with 5,000. By the time you get it spread all the way around, there's going to be a lawn of bacteria, a lawn of bacteria. By the time you get down here, you'll still get a couple of individual colonies. You can also use a loop dilution and a pour plate. So maybe you take your patient sample, put it in the broth, and you can pour it in on top of the agar, and it's going to get an even distribution. You're not diluting it all at all at this point, but you are going to get a lot of growth. Okay, but you can dilute it in a different way. So you have your original tube here, you poured it out, it grew a lot, you couldn't tell the individual colonies, so maybe you took a little bit of tube 1 and put it into tube 2 and into tube 3. You can see how the color is getting more dilute and more dilute. Then when you pour it on the plate, presumably you're going to get less bacteria on plate 3 than on plate 1, which means you could get discrete individual colonies that you could pick and use to study. Our final tool for inoculating a plate is to do a spread plate. So you use a pipette with your original sample. Maybe it's a water sample that you're wanting to test. You pipette it on top of the agar, and then you can use this glass rod instead of the loop. We refer to it as a hockey stick, and you can spread it all the way around, get it all nice and even, and then you can see what grows. This is often the tool used to enumerate or count the number of bacteria that we started with.